Thank you very much. I appreciate the welcome. So as Caitlin explained, I wear many hats. So I'm a doctoral student at FIT. I'm also a professor at EFSC, and I'm the director of science here. I don't like to be bored. But today's presentation, I'm wearing my FIT sorrel hat. So this was a project funded by Brevard County Save Our Indian River Lagoon, or sorrel program, in a partnership with FIT to do the research. Oh, I don't have the clicker. I'm like, Caitlin, move it. All right, so just a little overview. You are all familiar. There's an issue in the Indian River Lagoon, right? It's not as healthy as we want it to be, and we need to do something about it. So just to summarize, we have some main impacts that are causing what we call muck. So freshwater runoff seems like a good thing, right? We're in an estuary. We want fresh water and salt water to mix, and that is true. But there are such things as too much fresh water and too little fresh water in estuaries. In this case, we're worrying about too much fresh water. So rainfall, our river inflows into the lagoon bring us fresh water. We're also having things like runoff from people's yards from rain that carry yard clippings, grass clippings, everything into the lagoon as well. So what we worry about with that freshwater runoff is something called stratification. And so what we worry about with stratification is that our water column, instead of being nice and mixed like we want it to be, ends up in two distinct layers. And so at the bottom, our bottom layer is that nice salty water. It gets pretty saline. And because it's high in salinity, it can't hold as much dissolved oxygen as fresh water. And so the bottom layer tends to be lower in oxygen and more saline. The top layer is the opposite, right? That's where our fresh water is collecting. And because it interfaces with the surface of the water, we're getting lots of oxygen saturation. So lots of our oxygen is in that top layer, but those layers are stratified, they don't mix. And so our sediments at the bottom become an issue when we have low dissolved oxygen. We also have things to worry about like excess fertilizer runoff, grass clippings, pet waste. All of these things bring even more nutrients, nitrogen and phosphorus in particular, into our lagoon. And excess nutrients build up at the bottom and become muck. So the last thing I want to talk about that contributes to muck is our harmful algal blooms. And so these things are all intertwined, right? So muck is very, very fine grained. It can be resuspended in the water column quite easily. If you've walked through the water over here, it's less than a foot of water. You can't see your feet. We call that turbidity. It's the amount of sediment suspended within that water column. And unfortunately, high turbidity is detrimental to things like seagrasses. They physically block sunlight from reaching the seagrasses so they can photosynthesize. And then when we have harmful algal blooms, they're consuming the excess nutrients in the water column, again, physically blocking the light. And then unfortunately, they die, and then they themselves become decomposing material at the bottom of our lagoon. And so this is Jessica up here. She's holding a plate of muck from last week's sampling at Sykes Creek, and there's a reason she's leaning away from it, right? We all know that awful smell. It's not something pleasant to be around. So let's talk a little bit more about muck. So if we want to be really specific, muck is fine-grained, organic rich, polluted sediment. So, and when we say fine-grained, it is really almost minuscule. We have jars of it that we can bring out in the back that I worry that it's like a ticking time bomb. If someone drops it, that's the end. We don't want to smell that. We don't want that around. So it's also smothers our natural sandy sediments, right? So our estuary should have beautiful, clean sediments that are hosting seagrass throughout the lagoon. Unfortunately, we have a buildup of muck on top of those sediments that smothers seagrasses. And it also produces a compound called hydrogen sulfide. And that can cause lower dissolved oxygen levels at the bottom. So at the bottom of our sediments, our benthic environment, there's a lot of decomposition going on. It's crucial for the nutrient cycling in the lagoon. Now, decomposition requires oxygen to occur. And so when we have areas that have low dissolved oxygen levels, we also see a slow in decomposition rate. Okay, so, but I did mention it does produce that toxic hydrogen sulfide gas that smells like rotten eggs. On a bad day when it's really windy, you can smell it up here, not so great. And then the last thing I want to focus on is that muck, because it is fine-grained, tends to settle in low energy, deeper environments. And this is where we get things that are referred to as muck pits. So I'll show you in a later slide, but our muck pits where we're seeing almost 8 feet of muck being removed tend to be the deepest areas in the lagoon. Okay, this is the fun part. So benthic infauna are my area of study. So what are they? They are organisms that live in or on, so in fauna, the bottom of a water body, so benthic. And so this is a variety of diverse organisms: clams, polychaete worms, crustacean, amphipods, arthropods, tons and tons of diversity. And these things are crucial for our food webs. They are the very base of our estuary food web. So this is what a healthy benthic environment looks like. So up top is a picture of my microscope that we look at the sediments through at FIT. That is a beautiful, healthy sample. You can see different mollusks. There's some polychaete worms in there. And if you'll notice, it's not mucky. And then that is a picture of what we associate with those kinds of samples, where we have healthy seagrasses abundant. Okay, now the one thing I do want to point out with these guys is in a healthy environment, we have something that's called, well, in any benthic environment, we have something called the boundary layer. And it is a minuscule, small, thin layer right above the sediments that is stagnant while the rest of the water column moves. And so that boundary layer is really crucial for the sediments because it needs oxygen to pass through that layer in order for a decomposition to occur. These organisms make that happen. They move up and around that boundary layer, they disturb the sediments and bring oxygen into those environments. They are also the ones actively decomposing and they oxygenate the surface sediments for things like seagrasses. Okay, come on. Now let's talk about a polluted benthic environment. So up top is another, that's a sample under the scope. That is a polychaete worm. So this is Aleta succinea, and this is a pollution-tolerant worm that lives in muck pits. And surprisingly enough, we find it a lot in Sykes Creek. So in these polluted benthic environments, we expect and have seen very little to no diversity. So when we talk about our sample grabs, we grab an area of 225 square centimeters. In that area, we can expect in a healthy environment to have up to 50,000 organisms. In a sample like this, same area, you're expecting 0 to 5 organisms. So not great. So low abundance, low diversity, and the ones that are there are pollution-tolerant generalists. So our polychaete worms can tolerate everything. They eat the sediment, as you can see in this picture. His gut tract is just full of sand. They eat the sediment and they respirate through their entire body. The surface area of their body is how they respirate, and they have specialized gills. So they can get oxygen in areas that have very little oxygen. And so we see them in these unhealthy environments. All right, now this is a dredging project. So I just want to explain the difference between normal, everyday maintenance and navigational dredging to what we're doing, which is environmental and remediation dredging. So what you see every day in the IRL navigational maintenance dredging is exactly what it sounds like. We're scooping accumulated sediments and debris off the bottom to make it easier for boats to access and navigate. Now, environmental and remediation dredging is much more specific. We have target areas of specific sediments we want to remove, and our dredge barges have specific instructions of how much and where to remove. It also needs to amplify not resuspending the sediment into the water column, because as I said earlier, when we have that muck resuspended, that's like the perfect environment to have a harmful algal bloom start. And we don't want that when we're trying to help the lagoon. So we're minimizing sediment resuspension and collecting polluted and contaminated sediments off the bottom, not navigational. So Sykes Creek is environmental and remediation dredging. All right, so this picture is from Sorrel. Thanks you guys for providing this. It's just a visual of what our dredge barges are doing out there. So this is all you see when you're boating. But down below, we've got a very specific targeting arm that is gathering the polluted and contaminated sediments. That's pulling it up. And then that muck gets collected in a permitted dredge material management area where it is dried out and dewatered and then tested for contaminants. If it has contaminants like heavy metals, which is what we are primarily concerned about, it goes to a specific landfill. So we don't want it back in the environment, it gets placed in a landfill. If it's not contaminated, which most of this is not contaminated, we can reuse it for agriculture, which is a great use. Okay, so Sykes Creek is in the northern IRL. We're in the Banana River Basin, and it is highly residential. It's all homes, and all of these homes tend to have seawalls. It is the go-to method for armoring and coastal protection. However, it completely eliminates that intertidal habitat. So what we're seeing and what we saw last week when we went out to sample is that we have manicured lawns up to inches away from the lagoon. And so when you have lawn services coming to trim and fertilize your lawn, like we saw last week, they're fertilizing directly into the lagoon, unfortunately. Now, what I recommend as a living shoreline person is if you do have a seawall in your yard, to produce or to provide a natural filtration area. So foot in diameter, maybe a strip of natural vegetation that can physically filter out pollution and runoff from yards. And that keeps it from entering our estuary. Okay, so here's a look at our experimental site map. We have quite a few different kinds of sites for this project. So our SDS marked sites, which are primarily up in the left-hand corner here, are for seagrass monitoring. We know there has been seagrass there historically. In several of the transects, there still is. And so that research is looking at how neighboring dredging, because it's happening right next door, is going to impact those seagrass beds. Now, SMC sites are what we call our muck control sites. So these are sites that are not currently dredged, so that we can compare non-dredged to dredged to see how that works. And then we have our muck-edged transects. We have 3 of these. So if you look at our map, you can see the deep areas of Sykes Creek. And those are our muck pits. And so we do a transect line of sampling across those muck pits. So we take samples at the top, on the slope, and at the very bottom of the muck pits to see what their sediments look like. Oops. And then finally, we have the Sykes muck dredge sites. Now, these aren't muck pits. These are areas that have like 0 to 2 feet of muck that are still being removed, but are not considered muck pits. Okay, so this is our really fun. We do this quarterly and go out and get covered in muck. It's super fun. So primarily we do petite ponar grab samples, which is what is shown in the picture there. That's our lab mate Connor. So it's tied to the boat. We drop it to the bottom. It's like almost like a claw. It grabs a set amount of sediment. We pull it up. And for our sediment analysis, we don't do anything with it. We put it in a bag and then analyze it in the lab. For our infauna samples, we sieve it through 500 micron mesh to get rid of larger, or to get rid of some of the sediment, but not animals. And then we take that filtered sample and look at it under the microscope in the lab. We also take water quality data, and that is, again, to look at things like stratification and low dissolved oxygen at the bottom. So we take water quality samples at the top of our sample site and at the bottom. to see if they are drastically different and if we're seeing stratification. And what we do see is in some of our muck sites, they're almost 9 meters deep, which is crazy. So there is a difference between the top and the bottom. And so we need to know what those environmental conditions are like at the bottom when we pull that sediment versus what it looks like at the top. We also look at depth and turbidity. Depth is super important for our infauna. They don't tend to live at higher depths, so we don't expect them to come back in huge abundances at the very bottom of the muck pits, but we do expect to see some regeneration. Okay, so in the lab, I do sediment processing. So first thing we do is water content analysis. So it's simple enough. We spoon out a scoop of sediment, weigh it, then we put it in the oven for 24 hours and weigh it again and see how much water was lost. And that is considered its water content. Organic matter content is after water content. So we take that dried sediment out of the oven, we crush it up so it has as much surface area as possible. And then we put a set amount into a muffle furnace at 1600 degrees. And so that burns off any organic content in the sediment whatsoever. So then we weigh it again after it comes out of the muffle furnace and see how much organic matter has burned off in that process. And then finally, we analyze the grain size. This is important because we know that muck is super fine grained. And so we can build sediment profiles depending on their organic content, water content, and grain size to guide environmental dredging and restoration efforts. Now for infauna, it's a little bit different. You'll notice this is a picture of my station in the lab. So I've got my laptop up. We have a comprehensive guide of the species that are commonly found within the Indian River Lagoon. And so we identify any infauna we can to species, if we can, if not to genus, and then we count them. So you'll notice next to my microscope, I've got 2 counters. So I'm currently monitoring a site in Kennedy Space Center that was restored over a year ago and is doing fantastic. And the counters are necessary because when we get to 50,000, I tend to lose count. Okay, so this is what Sykes Creek was before dredging. So we have been sampling since 2020 on this project. Dredging was done last year. So we have 2 full years of free data. And what we're seeing is that our sediments are mucky. They have high water content, they have high organic content, and they have really low grain size. So they're exactly fit that profile of muck. Our infauna fit muck too. We have very low abundance, like I said, 0 to 5 versus 50,000. And the species diversity is little to none. It tends to be those polychaete worms that are able to tolerate low dissolved oxygen and pollution. So in this graph here on the side, we have organic matter content percentage at the bottom. Highest percentage we usually see is about 27%, which is mind-blowing. Healthy estuarian environments have up to 5% of organic matter in their sediments. So 27% is like, so with a decrease, with an increase in organic matter content, we have a decrease in abundance of benthic infauna. Makes sense. They can't tolerate those sediments. And in Sykes Creek, our muck pits, our 3 muck pits, were right around that 20 to 25% range for organic matter content. So very mucky, makes sense why we don't have infauna there. All right. I know this is, it turned. Okay, well, it's supposed to be that way. I know it's difficult to see, but this is the dredge plan that was provided to the county. And so in the more pink areas, sorry if you're red, green, colorblind, is where they have dredged. In the darker purple areas is where they plan to dredge that hasn't been dredged yet. But what I wanted to point out on this map is that this is our DMMA. That's our permitted dredge material management area, where all of that the sediments taken off the bottom rests to be dewatered and dry out. Now this is the one, okay, good, that's the right direction. This one is what I want to highlight. So this is from the dredge contractors saying how much they have removed from these sites. So I know you can't read it, but the purple areas are 0 to 2 feet removed of muck. Dark blue is 2 to 4 feet removed. Light blue is 4 to 6, and green is 6 to 8. So you can see where our muck pits are, right? Yeah, so in our muck pits, we did have 6 to 8 feet of muck removed through this environmental dredging. Please, there we go. So it's important to note that we've only analyzed one post-dredge sample since. We went out last week and collected, that data has not been analyzed yet. So we do only have one set of data to post-dredge to kind of base these initial conclusions on. We'll get more data, don't worry. So what we're seeing though is encouraging, it's really encouraging. So we're seeing lower organic matter content in those muck pits, where we were seeing 25 to 25% organic matter, we're now seeing 8%, which is a huge increase. And with encouraging decomposition, that should continue to drop. So hopefully next time we go out there, or when I analyze this last week's data, we'll see some lower organic matter content there. We're also seeing larger grain size, which makes sense because their goal was to get it down to that sand, which is larger in grain size. So they achieved their goal. And then our infauna have both higher abundance and higher diversity, which is fantastic. So in this picture here are some of the species that we have seen in our post-dredge samples at Sykes Creek. So we've got foraminifera, amphipods, annelid worms, so polychaetes, and bivalves, all sorts, much more diversity. Especially what's really encouraging is the top row, the middle, he kind of looks like a tank. So that one's O. Smithy. That one is specifically been published on to be associated with seagrasses. So we are excited to see him. They are not pollution tolerant whatsoever. So it's very encouraging that he's there, and he's not the only one. So very exciting. So what's next? I get to keep scooping muck off the bottom of the lagoon, hopefully less and less muck as we go on. So again, we want to have as much post-dredge data as we have pre-dredge data, or more if we can. So we're going to be analyzing sediment, infauna, and water quality throughout all of these sampling. We're also going to analyze how that community structure responds to environmental dredging, because we know the basic sediment profiles of healthy benthic environments. And these guys are never, this profile is never going to match perfectly what a healthy benthic environment that's never been disturbed looks like. But we can restore it to a level of diversity and abundance that has the same ecosystem services, which is what's really important. So this is the portion that feeds into my dissertation. So my dissertation is looking at different types of restoration and how they impact the benthic environment and the things living in it. And so this is a very important project for me. I really enjoy it. And then our lab mate, Sean, is a seagrass guru. And so he's taking over those seagrass sites and expanding on that data set to look at how neighboring environmental dredging affects seagrass beds. All right, one huge thank you to Brevard County Sorrel Program. They provided all of the funding for this project and have been incredibly supportive. So thank you, Sorrel. And then, come on. Oop. Thank you to all of my lab mates and professors. So we have a huge group of FIT students working on this. Dr. Austin Fox, Dr. Kevin Johnson, Dr. Emily Ralston. This is Jessica up there. Dr. Fox is next to her. Yes, he is wearing a pirate hat. He puts fun into sampling. Mary McDonald and Rebecca English with the Fox Lab. And then my PhD colleagues, Sean Crowley and Connor Wong, are also huge influences on this project. So thank you very, very much. We couldn't do it without you. And I did want to include this because Sorrel put up a really, really nice informational sign at Kiwanis Park about the project, and it's there, and it's really good. So go see it. Thank you. I would like to answer any questions, please.
Thank you so much. So I'm going to come around and collect the note cards from our in-person audience. To get you started, though, We've got a couple questions coming through Zoom. The first one is, are there any plans for Merritt Island National Wildlife Refuge to remove muck from their impoundments that flow into the lagoon? Their impoundments flow muck through culverts directly into the waterways.
That's a great question. Hopefully, yes. I am not the person to ask.
Okay, we've got one more question then over here. Is benthic infauna sampling expensive? Is it being utilized as a metric of success for dredging projects?
Great question. So no, it's really quite inexpensive. Our main gear is catering trays, thieves, and then the petite ponar grab, which is the most expensive. It's about $1,200. So it's very inexpensive to do. In terms of monitoring other projects, I'm not sure if benthic ecologists are involved. As far as our lab goes, no, we're not involved in any other dredging projects, but I'd love to be involved. So if you know of any, let me monitor it. I'd be happy to.
Thank you. Do you monitor the dewatered water?
No.
What are you doing?
I can answer. So this is Abby with Sorrel in the back there. She knows way more about the actual physical process of the dredging than I care to.
Yeah, so we, the county and our engineers that help the projects, Taylor Engineering is here, they've worked on the project too. We do test that material. But something that the county added to these projects, it's not required by permits, but Florida Tech researchers found that the interstitial water has high concentrations of nitrogen and phosphorus. Because of this, the county kind of challenged the dredging industry to get creative and come up with innovative technology to treat this water before it's discharged. So we're actually removing the nutrients from the water itself, and then it's returned to the lagoon. Yep.
Yeah, it's a complicated process that a lot of science has gone into. So
yeah, and so this extra step has helped us remove thousands of extra pounds of nitrogen and phosphorus before the water is returned to the lagoon.
The facility there up in Concord.
That is for our Grand Canal, an unincorporated satellite beach. That is the upland dredge material management area. Yes.
Okay. All right. How often should this be repeated?
Sampling, monitoring, dredging. That's a great question. I don't know the answer of that, mostly because it depends on the rate of the sedimentation of the muck. So we'll see how that comes back over time, I would say. Abby's got an answer. We'll leave Abby with a mic.
Austin Fox from Florida Tech has helped the county a lot prioritize the muck removal sites. With the Sorrel plan, every year it gets updated and it runs through a citizen oversight committee. We get a lot of different input, and then it's voted whether or not to add a project to that plan. And so we already have 21 funded environmental dredging projects approved in our plan. And those are based on the sites identified by Help with Florida Tech with the highest nutrient fluxes, and also taking in the logistics. And so we're kind of prioritizing them with mixture of the science. So what pits have the highest nutrient fluxes, and then also what's available for land. We do need upland dredge material management areas to help dry out this sediment.
And when you're removing 8 feet from a small area, it really adds up in volume.
Yeah, and so one thing that we like to say is the trash can is full. And so Mara was talking about, these deep pits, they're filling up to the top. We want to help remove that sediment before it flows into those nearby healthy adjacent sandy areas. So we're kind of following through our projects with a plan with no intention to go back and redredge those sites.
Yeah, hopefully there's not a need to redredge them.
The land that you guys are putting the sediment on in Sykes Creek, the island.
Yeah.
I mean, I've walked it as a child. Do you have room there, or is the property that's surrounding Esther with Brevard, that Brevard County just purchased for the property appraiser's website is what I'm kind of doing some research, but I mean, the land is completely cleared. Is that the new site for the Sykes Creek muck?
Yes, very good question. You're correct. So we kind of internally phase our projects to help us out. So phase 1 of Sykes Creek, which is what they've been monitoring, was complete. They removed about 76,000 cubic yards of muck from that east area. And so all of that is on that island and it's being used, it's remaining on that island. It's been tested for, and then that island, there was a bunch of Brazilian peppers and stuff. So right now they're working on restoring it, planting natives, getting the slopes correct and making sure that it's actually kind of better than how they found it. And then for phase 2 of this project, there is a lot more material located in those western canals and kind of adjacent open areas to the wildlife sanctuary. And that material will be pumped to the facility that is being cleared. We call that the Nevada, the Walmart site.
Yeah.
So they are
yeah.
clearing that. And that will be a temporary dredge material management area for this project. And so the material will go there just to get dried. It's not going to stay there. It's
Right.
going to get trucked off to a permitted disposal facility. And that part is left up to our contractors to figure out. So they're the ones with the relationships.
Okay, what's your plan with the property? What's the county's plan of the property in the future?
It'll be retrofitted as a stormwater treatment site. So it'll be a green land, green space with a stormwater pond to help filter stormwater before it returns.
Great.
There's a lot of controversy,
Always
through
is.
social media,
Yes.
at the Triangle. I pass that site every day, 10 times a day.
Yeah. So we just had a sign, the same sign that she showed on her slide, put up in front of that site.
Yeah.
So hopefully people know it's related. And so, yeah, it was unfortunate. They did have to remove some vegetation from there. We did leave a 20-foot and 15-foot
Buffer zone.
buffer, yeah.
Buffer zone, yeah.
Buffer site, and so, but a lot of that internal stuff was Brazilian peppers. We followed permits. We had to do gopher tortoise surveys.
Yeah.
I'd like to spread the word.
Yeah, no.
Yeah,
appreciate it.
absolutely.
No, it is scary. It does. It is really scary.
But if it's beneficial for the waterway.
Yeah, so it's being built as a temporary dredge material management area. And the material is not going to stay there. It's just going to be dried out there and then the contractors will dispose of it. And then once the dredging projects are done using that site, it will be designed as a stormwater treatment pond.
Great.
So
Okay.
multi-use.
Did this dredging project have any impact on manatees? Is there any time that you could not dredge because of manatees?
Yes, when you're working in canals, you do have mandated closure dates. It kind of depends on what the agencies focus on. So for example, our Grand Canal project in the unincorporated Satellite Beach area, they do have manatee closure dates from December 1st to March 15th. So we're just about to start that
Yeah.
project back up. And then for these areas like Sykes Creek, there are manatee closure dates that you have to oblige to, but in the canal system. So
Yeah.
during those manatee seasons, they can move into those open areas that were shown on those maps, those deep areas where the manatees won't go. So they do. And then they have
Yes.
designated
you
manatee
go back to the
rivers,
map?
so they are supposed to shut down.
The first map with the light pink.
Let me
I
bring the
can't- I
microphone
can't
back to you.
control
Hold
it.
on 1 second. Bringing the microphones, yeah.
Where does the funding come from? This was a grant or this project?
So Brevard County Sorrel Program is funded through the half-cent sales tax.
I live on a canal that's a. not
Yeah,
a
this one.
navigable canal, but everybody has a dock.
Yep.
and it's runoff from the street. And so we have so much sediment in there and muck,
Yeah,
where our water level is about 2 feet and it's another 4 feet of muck.
yeah.
We have a real difficulty of who can fund to do it.
That's a question I get all the time, especially when I talk to homeowners about softening previously hardened shorelines like seawalls, is where does that money come from? That's a great question. FWC has a program for private homeowners that they have allocated money set aside to work with you guys. So I have a project going on in Malabar Rocky Point Road, and FWC, their private homeowner program has stepped in because it's going to cost a lot of money. And they're finding this funding sources. So there are organizations and people out there like myself who can help you find grants and get started on projects like this. It's tough to get started. Once you get over the hurdle of money and permitting, then it's quite easy. And this is something I do on an everyday basis. So please let me know if you have specific projects you want help with, or just kind of talk back and forth. Yeah.
This question is more of a comment. It says your muffle furnace at the current temperature might affect CaCO3 content and change
Calcium
perceived
carbonate.
organic content.
So what Our methods are cited from USGS for the muffle furnace. I know it gets up to 600 or 1600 degrees, but I don't know if that's where, I think it levels out at 1200 degrees and stays there for 4 hours. Yeah.
Thank you. What is the boundary layer caused by?
A lot of things. Primarily just the hydrology of the water. As it interfaces with the sediment layer, that benthic zone, it just naturally causes a kind of, it's almost like a centimeter thick of stagnant water. That's what we call the boundary layer. It's a natural phenomena, and that occurs in the ocean, estuaries, ponds, everywhere.
What is the technique for finding the top of a muck layer?
That's a good question. I don't know the answer to that one. Do you know how to find the top of a muck layer? Abby, we should just put Abby up here.
So I worked with Austin Fox at Florida Tech for about 4 years, and so it really takes just a lot of probing over and over and over again. And so when he was teaching me, you have to feel for the pole.
Yeah.
We use a PVC pole because some people use like metal poles, but it's hard to feel it go through that top layer. So you can kind of feel the restriction, but you have to just keep practicing. So every time I
It's
would
a
come,
feel.
Dr. Fox would come next to me and he would see it and we would see, make sure it's lining. And then another way you can do it is what you mentioned was lowering a Secchi disc.
Yeah.
It's just this round, thin disc, and you can lower that down and that surface area will kind of get caught on the top of the muck.
Yeah.
So that's another way you can validate that you've reached that surface.
So that's
It is
how
difficult though.
it is. So that's how we determine depth in our sites, is using a Secchi disc. So it's the feel of the pull on the rope when you know that it's hit the muck layer. It's just something I've learned over time and that I've refined. So it's practice.
Seems like really simple thing to do, just stick a pole down there.
Right.
But it actually does take a lot of practice getting the feel for what that resistance kind of
Yeah.
feels like. And Austin Fox really has it down.
Yeah, he does.
Alrighty. What is the cost for each of these dredging events?
That would be an Abby question. I just do the
So,
monitoring.
If you visit the Save Our Indian River Lagoon website online, you can see our 500-page plan that documents exactly where all of this funding gathered by the half-cent sales tax is going. So we have a breakdown of project costs, what we anticipate it to cost, what past have costed, and then all of that is detailed within that long plan.
So read 500 pages.
But no, you can go down, it's like table
Yeah.
431.
Oh, it's very well laid out. You can
Yes,
find the
and you can
specific
see
section.
exactly what you estimate the cost to be on. Obviously, we had changes. If you have a project go out to bid, 6 years ago, construction costs have gone up. So
Yeah.
you're going to see some fluctuation. So it's our best estimate that we have based on previous projects that were completed and looking at those costs.
Aren't we somewhere in the neighborhood of what, 35% of the total historical funding is going to mud?
Yes,
It started
and
out
interstitial.
6 to 8 years ago at like 65%.
Mhm.
Yeah, so they really cut the funding down for that, and there's a heavy focus on stopping the pollution before it enters the lagoon, stopping the reaccumulation of muck, while also working in conjunction to remove what's already existing there.
Yeah.
They are not cheap projects.
No.
It's a lot of manpower, it's a lot of testing, it's a lot of trucking. They're definitely very complicated and complex projects.
Broadcast that message, because until we adopt low impact development, keep the water where it falls, we're just going to have this question earlier about recurrence of muck once it's been dredged. We dredged Turkey Creek in 95, 20 years later, we were dredging again.
Right.
Yeah, so I do think that all of us, we definitely, all individuals have our role to play, bigger entities have their role to play. And if you are interested in kind of figuring out what you can do to help prevent the muck accumulating, you can join Lagoon Loyal online, and it gives you a bunch of tips and tricks from at home, what you can do to benefit the lagoon. And in return, you'll actually get points that can be used as coupons around the area. So it is important to address what's coming in and also focus on what's coming out.
Yeah. We've got
I'm going to
hands.
bring the mic.
Just ask if you'd be able to touch on any of the projects or policies that are being implemented to mitigate the reintroduction or reaccumulation of muck.
Do you want me to give Abby my microphone?
Like LIDGSI or like what specifically?
With the Safer Indian Lagoon program, there is a range of projects that they're focusing on. So subject to sewer conversions and upgrades, testing leaky lateral pipes, wastewater treatment facility upgrades, oyster restoration, seagrass restoration, living shoreline,
baffle
stormwater
boxes.
treatment, baffle boxes. There's a whole variety of projects because it's not, there's no one silver bullet.
Yeah.
We're not saying dredging is going to fix the lagoon. It's
Right.
a multifaceted approach. Then we just have to keep going. Yeah,
Effective it is.
it is.
Like how resilient our ecosystem.
I do think, if we
Yeah,
give the environment what it needs to be happy, it'll do what it needs
yeah,
to do to repair itself.
it will naturally repair itself if we give it the option. We just can't keep pouring muck back on it, So yeah, our results are encouraging. Again, one data set post dredge, but we'll see. Hopefully, give me a year and I'll do it again and I'll give you an update.
I'm going to continue to stand next to Abby for this.
I think this is the camera area.
When do we vote again for Sorrel?
Oh, this is an important question.
Okay, I know the answer. I'm pretty sure they're trying right now to get it on the ballot for this November. Originally, it was going to be in 2026.
Yeah.
But right now, the Brevard Indian River Coalition is working to get it on the ballot.
Yeah,
for
so
this.
they are hoping to get it back on the ballot to be re-upped. So please pay attention to that and vote accordingly, please.
With the widespread use of herbicides along and in waterways by FWC, the Army Corps of Engineers, and cities and counties, how much impact does this have on muck accumulation with dead grasses?
Herbicides?
Yes.
So I don't have a quantifiable answer for that, but there is obviously an impact of herbicides, right? It's killing vegetation, and what muck is decaying plant material and other stuff that has accumulated. So that's just adding to our muck problem. Again, it's one of those things of how do you mitigate that behavior when the herbicides are being used for a purpose and then they're being negatively impactful on another purpose. It's a kind of a jungle to kind of navigate through, but it's one of those things where we have to find the fine balance of, yes, you can use them to this amount. So when we talk about nutrients and fertilizers, we're always talking about it in excess. There is an acceptable amount of use. It's when we exceed that amount, it becomes extremely harmful. And right now we're exceeding that amount on all fronts. So.
Thank you.
Mhm.
And then this question is just asking if you could go over one more time how you currently monitor changes regarding biodiversity in your examined areas.
Oh, sure. So for diversity, so it's a really enormous Excel spreadsheet. It's pretty impressive. So what we go through first is abundance. So we go through and we physically count everything in our samples. And so we can extrapolate that and to fit a broader area. So what we report on is per 225 square centimeters. That is the area of the Ponar grab. And so the numbers I'm presenting is per 225 square centimeters. And so what we can see over time, because we have a set area, is if we have a physical increase in numbers, that's an increase in abundance, right? But with diversity, we do an analysis called the Shannon-Weiner Index of Diversity. And that is a statistical analysis that compares how many species you have in one site to another, or how many species you have in one site over time. And so you can see a statistical significance in increasing diversity in our post-dredge sites.
Thank you. And that is all of our questions.
Yay!
Thank you so much, Mara. Let's give her a big round of applause.
And Abby.
For answering questions. Thank you all so much for tuning in.
Well, give